Hello, my name is Younus Zuberi.
And I am Sabrina Portilla.
This is our presentation on antibiotic sensitivities of planktonic and biofilm cultures. Bacteria are found in two growth modes, planktonic and biofilms. Planktonic bacteria are free-floating individual bacteria that can be found in places such as running water. Biofilms are communities of bacteria that can be formed on damp surfaces. In nature, they are found with more than one species. They also produce extracellular polymeric substances which surround the community and help them be more resistant to environmental stress. Biofilms and planktonic also differ genetically. The purpose of the study is to test the effects of the antibiotic kanamycin on mixed species cultures of E. coli and Serratia marcescens in planktonic and biofilm states.
Materials in this experiment include the bacteria E. coli and Serratia marcescens. They are cultured on tripixel agar plates and then grown in as a liquid culture to reach their log phase, where the bacteria are actively dividing. This is the most effective phase to use antibiotics. Kanamycin was used to perform the minimum inhibitory concentration and minimum bactericidal concentration in planktonic, while in biofilms we use the crystal violet colorimetric assay in place of the MIC. We also do differential tests like eosinomethylene blue and phenol red tests to distinguish bacteria. Minimum inhibitory concentration is the concentration at which the antibiotic stops the growth of bacteria. We set up concentrations of antibiotic from 0 through 1024 micrograms per milliliter, with E. coli and Serratia into all of the tubes. As shown in Figure 1, our planktonic results for the mixed bacteria show growth in tubes with 0, 2, and 4 micrograms per milliliter, where the bacteria was not inhibited. The first clear tube shown here is 8 micrograms per milliliter, and that indicates our MIC.
Figure 2 shows the minimum bactericidal concentration, which is the lowest concentration at which the antibiotic has killed all the bacteria. Our test showed that the MBC was 512 micrograms per milliliter. Table 1 has data from single-species planktonic, MIC, and MBC cultures, which show that both bacteria were inhibited and killed at 2 and 4 micrograms per milliliters respectively. This shows that even in planktonic form, the multi-species culture is more resistant to the antibiotic.
To conclude, our results from planktonic culture show Serratia to be the only bacterium present throughout higher concentrations. However, both organisms, E. coli and Serratia, are shown to be present in the lower concentrations of the antibiotic.
We have also determined that the mixed species planktonic culture, the MIC is 8 micrograms per milliliters, and the MBC is 512 micrograms per milliliter, which are higher than the individual species culture. Using the methods mentioned, we are currently running tests to obtain the results for biofilm cultures. We have also considered future directions for the project, where we can test different stressors, like bacterial viruses known as bacteriophages. We have also considered adding another organism to see if the interactions will be different. Thank you for listening.